Hi, I'm Isabella, and this is the One to the World Slime Mold Open Inquiry Lab. So for a little bit of background on slime molds are considered protists, and they have more than 900 different species. Slime molds are also just not one organism or amoeba. There are thousands of them bonded together, and they work through problems together, such as the maze in this experiment. They also reproduce by dividing into two, which is apparent in their branch-like formation that you'll see. So right off the bat, you can tell that slime molds are super interesting to look at, but what's even more interesting is the amount of intelligence that they actually seem to possess. In our classroom experiment, we watched the slime mold navigate its way through a maze to seek out food. While working with slime mold for this experiment, my group and I gained a lot of knowledge and a better understanding of how slime mold works. So we began our experiment with a sterile petri dish, and then we constructed a maze that would fit inside the petri dish, and then we had to sterilize that, and all sterilization was done with isopropyl alcohol. So then we used a sterile pipette to cover the bottom of the dish, the inside and the outside of the maze with agar. Then we waited for the agar to solidify. We proceeded to use sterile forceps to place two pieces of paper containing slime mold culture on opposite sides of the maze. Then we placed a sterile oat on top of each paper and moistened the oats with sterile water. Then we sealed up the sides of the petri dish with parafilm and then proceeded to cover it with tin foil so it stayed in the dark. This gave the slime mold ideal conditions for growth. Then we left the slime mold to grow for 24 hours. This was the slime mold's growth after 24 hours. You can see that there was obviously a lot of growth immediately. After observing the slime mold again at about hour 33, it still appeared to be growing fairly quickly. Notice that the slime mold did find its way through the maze to its food sources and even went underneath the maze. This is what the slime mold growth was at about 48 hours, and it's most noticeable around the outside of the maze. These next pictures are of the slime mold throughout the next few days. The amount of slime mold is still increasing because the food source is still being used up. I forgot to mention it earlier, but as you can see, one of the oats moved from its original location when we first prepared the petri dish to outside of the maze. This happened in my backpack because I was carrying it around and I didn't think it would move, but it did. But as you can see, the slime is still moving towards the food source, and there is less in the middle because it's already used up that food source, and now it's going to the food source on the outside. You can see here that the general amount of slime mold and slime mold growth has started to decrease because of the dwindling food source. However, the slime is still spreading itself out throughout the petri dish. At this point, it was clear that the neon yellow slime mold had not shrunk back from the dead ends, but had gone under the Legos. It had also connected the two food sources, doing so in the shortest distance possible. It was also clear that slime mold navigated a maze almost as if it had a brain. After a week of being on vacation, I picked up my slime mold again, and it looked as if it had died. I did further research and found that some of these amoeba-like organisms become spores and survive to the next generation, while other cells become stalks and die. For the second part of this experiment, we repeated the same steps to set up the maze in the petri dish, and then we added the agar again. At this point, had I not forgotten to bring the previous part of this experiment to school, we should have sterilized our maze, gotten a new petri dish, filled it with agar, added the same oats from before with the live slime mold on top on opposite sides, and then add two new oats on opposite sides as well. However, in order to accommodate our circumstance, we placed two new slime mold culture papers on opposite ends of the maze, along with sterile oats on top of them. We moistened the sterile oats with sterile water again. On the other end, we placed two new oats as well, but for the open inquiry part of this lab, we decided to moisten them with blue and yellow food coloring. Then we proceeded to wrap it in parafilm just like the first one, and then we waited 24 hours. At the 24-hour mark, I found that the slime mold had not grown as quickly as it had before. This was the slime mold at the 33-hour mark. It was still growing steadily, but not quite as fast as the first one. The next few days showed that the slime mold was still growing and began to grow more rapidly, but we came to the conclusion that the food coloring probably wasn't affecting that. However, there appears to be one small physical effect of the food coloring, and that is if you look on the outside of the maze, the slime mold actually appears to be more green rather than yellow. Nevertheless, this slime mold still connects the two food sources in the fastest way possible and finds its way outside of the maze. Keep in mind the results may have been drastically different if I had remembered to bring the other part of the experiment and use the older experienced slime in this maze. Further research shows that slime molds are smart enough to lay a trail to find their way around mazes. Not only do they learn these mazes by navigating them with slime, but when one piece of slime mold was added to another that had not previously completed the maze, the experienced slime molds seemed to teach the others and they would complete the maze faster than before. Thus, with the presence of experienced slime mold, the maze may have been completed noticeably faster. In conclusion, this lab indicates a high intelligence in slime molds. Additional facts about slime molds are their ability of chemotaxis, moving towards or away from different chemicals which they sense in their environment. They are also healthy eaters. They normally go for a diet composed of 2/3 protein and 1/3 carbohydrates. This is why we used oats in this experiment. It is truly incredible just how easily they can navigate a maze. There's clearly a lot more to learn about these complex organisms.